So now let me pass to our next speaker from the Mumbles project. This is Professor Gilles van Wezel from Leiden University and the coordinator of the Marbles project. Okay, so dear Gilles, are you here? That is great. You are sharing already. Okay, so we can see your presentation. Great. We don't hear you, I think.
Can you hear me now?
Yes, loud and clear.
Very good. Okay, that's great. Okay, thank you very much, Kostas, for coordinating this. So
Thank you
let me
very
see
much
where I am.
for being here.
Okay.
So the floor is yours, yes.
Yeah, thank you very much. So it's a great pleasure to present to you the Microbes consortium. Thank you very much for putting this meeting together between the 4 consortia. Our project is, as already said, we work on marine bioprospecting, and MARBLE stands for Harnessing Marine Microbes for Drug Discovery and for Sustainable Production of Fish and Crops. I'm going to do something that I don't normally do, so if I fail, then please bear with me. But we have a little promotional movie for 1/2 minute, which I'd like to share with you. I'm just going to see if that works. So I probably have to stop sharing that one, and then yeah. So can you see this? Okay.
Yes, we can see the video. Yes.
I'll share the movie. It's just 1/2 minutes.
Sure.
But
communities that live alongside marine hosts. These interacting communities, or microbiomes, are found in most marine life, including sponges, algae, and fish. Microbes keep plants and animals safe and healthy by producing biologically active compounds, which help protect their marine hosts from diseases. Researchers are authorized to sustainably collect samples and study these microbiomes and their biologically active compounds so they can replicate and use their properties to create eco-friendly natural substances for use in agricultural, aquacultural, and pharmaceutical products. Many modern medicinal drugs come from plants and microbes. The marine environment remains a largely untapped and underexplored source of such natural products. Finding and identifying bacteria, extracting ingredients, and meeting regulations make developing these products difficult. The world is facing a growing population and climate and biodiversity crises. In this context, the Marbles project is harnessing the power of marine microbiomes to try to find solutions to the challenges these crises bring. The project will create eco-friendly alternatives to the synthetic chemicals currently used in pharmaceuticals, agriculture, and aquaculture. The natural products created will make fish production more efficient and will support more sustainable crop production. Sometimes, to think big for the planet, we need to look at the small stuff. Follow Marbles to see how the harnessing of marine microbes can protect our oceans, food, and health.
okay, I think I should try and share my where am I? Share my presentation again. Okay.
Yes, great. We can see the presentation. Yeah.
Okay, thanks. Okay,
Thank you, Professor.
Yeah, cheers. So the overall aim of the project is to harness the unexplored potential of marine microbes as produce of antimicrobial agents and also as bioprotectants for aqua and agriculture, using ecology-based strategy for bioprospecting. The objectives of Marbles are to use microbes and microbial consortia, so I'll explain those later, to be used as bioprotectants. To find small molecules with antifungal, antimicrobial activity, which can be either be used for protecting marine sources, but also for humans and animal applications. Find elicitors that activate silent gene clusters for bioactive molecules. Find product candidates with potential human health applications for commercialization post-project, like environmentally friendly alternatives to antibiotics in agriculture, reducing AMR problems, environmentally friendly alternatives to synthetic chemicals in agriculture. We'd like to provide science-based advice for important ongoing policy discussions related to bio-based biotechnology. and communicate the findings of marbles to our stakeholders, obviously, and engage the public in advances in microbial biotechnology, and very importantly, I think, all aspects of biodiversity. So we have 14 partners from 10 countries. So the coordinators of the 7 work packages, well, we are work package 8 as well. The coordinators are Leiden University, also work package 3. We have University of College of Cork in Ireland, DTU in Denmark, Aberdeen, and you just heard Massa Jaspers, Fundación Medina in Granada in Spain, Absinthe, and Erin, who just produced that fantastic movie I just showed you. Another partners are among others the Naturalis Biodiversity Center, Fraunhofer, the SME NICONS, SINTEF, an organization in Norway, the ETH in Zurich, Euro Open Screen, and Wageningen University. Yeah, I won't go into detail, but this is how the consortium sort of collaborates. You can see that a lot of expertise which overlap. So several of our partners have led major EU projects before. So there are many EU and ERA projects that Marbles connects to. I'll share this presentation with you so that you can access this information at your own leisure in your own time. The consortium coordinate, it also connects closely to a panel of industry experts, what we call the PI, and advisory panel on policy and legal experts, which we call the APPLE. And yeah, with those partners, we'll make this project hopefully to a great success. Okay, briefly, I'll explain it a little bit later. So what's the Marbles concept? Well, the concept basically will look into microbes as nature's medicine makers, and how can we harness their power actually to not just find new molecules, but also find microbes and microbial consortia that we can use for host protection and for disease suppression. So we'll go for work package 1, where we essentially look at the, mind the biodiversity, obviously in a sustainable manner, in a really close, connection to the sustainable development goals, to make sure we get it right. We find new strains of microbiomes. Then the second part will sort of zoom in on that, as it were, to find so-called disease-suppressive consortia with some microbes like, probiotics in humans, which can help us suppress disease. So we'll move into disease-suppressive consortia. We'll also go into work package 3, where basically we start zooming in on the microbes that produce those disease-suppressive molecules, and we'll also find natural molecules, natural signals that in the niche, and I'll explain it in a minute, that in the niche would activate the production of antibiotics and antifungals. So these elicitors are also compounds, but they're not antibiotics, but they're molecules that we use to activate the production of antibiotics, for example, during high throughput screening. We then move to screening and validation. So work package 4 is to find those small molecules and generate chemical diversity in libraries. We go to compound elucidation, refinement towards novel bioactivity, and in work package 5, it's dedicated to screening and validation. Okay, so work package 6 will be dedicated to societal and valorization aspects, which is led by Absinthe, and work package 7 is all about dissemination, communication, and IP, and knowledge transfer, and stakeholders. And finally, work package 8 is the project management. Okay, so brief, just a few slides of a little bit, let's say, an intermezzo slides, as it were. So what are the concepts that are behind Marbles? So where do we start from? So we start from the concept that microbes are everywhere, right? So microbes protect, they're nature's medicine makers, as already said. So in nature, they protect animals, plants, us, fish, bees, whatever, from microbial protection. And this goes in a very close kind of association between microbes and their host. And that leads to what we call the cry for help concept. And that is an important aspect of Marbles as well. So the microbes live in close association with the host, like in this particular case, a plant. The microbes feed on natural polymers, polysaccharides that they can eat. But when the plant gets stressed, for example, by pathogenic microbes or by helminths or insects, the plant in turn sends SOS signals to the microbes, which elicit the production of the right kind of natural products that they need, be it antibiotics, antifungals, anti-insects, anti-helminthics. And these elicitor molecules, if we know them, if we find them, we can use those in high throughput screens or revitalize the screening with our partners like Dicons and Medina. Okay. So here's work that we call so suppressive and conducive soils. So these are the same plants growing in the same soil. In one particular case, the plant will be healthy. In another case, the plant is diseased by a fungal infection. And what's happening here is that microbes are obviously suppressing the disease in the top picture. So this is a simple example of something that we've done in the lab recently, which is just one example to show the power of microbes. So on the top left of this example, you see a plant grown on its own. On the bottom right, you see a plant with a Streptomyces bacterium. On the top right, you see, well, you don't see it, but believe me, there's a Streptomyces. And in the middle is a pathogenic fungus. And the fungus overgrows this plate. So it overgrows the plant. It overgrows the Streptomyces, but when you grow the two together, the fungus doesn't get near it. So what's happening here that we've now established that the plant is inducing the production of the antifungal in the Streptomyces. Okay, and then the Streptomyces kills the pathogen. And meanwhile, the Streptomyces actually induces the immune response in the plant. And this is a typical, these kind of examples we now seek to find in the marine biosphere. And there we'll also focus on drought stress, on salt stress, besides the microbial stress. And in terms of collections, we have collections already in-house. So we have our microbial collections, which either come from our partners in industry, in the SMEs, and also in the academic setting. We look specifically at the moment already extensively at sponge microbiomes. We obviously have metagenomes who will create those, and will further delve into the collections that we have from all the different partners, from both marine and, where necessary, also from terrestrial sources. Okay, so work package 1, like I said, we start looking for novel bioresources and to find approaches for biodiversity mining. And primarily, we focus here on sponges, on microalgae, and on the fish microbiome. So we harvest samples, we isolate strains and obtain also the metagenomes of these samples. And then using a novel platform consortia cultivation, we create the marbles strain and a microbial consortium collection that we're building at the moment. And finally, then we have the consortia and strains and extracts, which will then be generated in work package 1. Like I said, we then zoom into disease-suppressive consortia. So there's work package 2 to engineer and phenotypically profile marine microbiomes and the bioactive molecules. So we investigate, we interrogate the metagenome data and the microbiomes, phenotype and isolate growth promoting microbiomes and microbiomes that are disease suppressive, which can then already be screened in both work package 2, but also primarily in work package 5, which is our screening work package. Design first synthetic microbiomes and then test those for their beneficial properties in terms of protecting hosts in aquaculture and protecting plants. We then move to work package 3. So there a major question is, how do you prioritize the extant biosynthetic potential? And this slide is just to show you, just give you an idea again of the extent of what we're looking at the moment. So some 20 years ago, we started sequencing bacteria that produce antibiotics. And right now in the database, we already have more than 1 million biosynthetic gene clusters, or BGCs. And where do we start, right? If you have 1 million gene clusters, so which gene cluster do we prioritize? How do we know where to go? And that's incredibly difficult. So the approach we follow with work package 3 is like an omics-guided approach to awaken cryptic gene clusters and find the molecules produced in work package 2, let's say the protective molecules, and start working there before we take those further to work package 4 and 5. So I start with, among others, embedded genomes and the strain collections. And then we are developing elicitation approaches. So we look at the regulatory networks as control antibiotics. And once we know what the regulatory networks are, we can interfere with them, right? So like is shown here on the top picture, I have a Klebsiella strain, and we have 24, in this particular case, streptomycetes that are grown on agar plate. And you see there's just 4 of these produce a minute amount of antibacterial activity. But when we use the right elicitor, you can see get huge activation of antimicrobial activity. And knowing these kind of molecules, finding new signals that can activate these clusters is important to improve and revitalize our screening. So once we then zoom in on those BGCs and find a few candidates, like indicated here, we can then analyze those further in work package 4 and find the lead candidates that we get out of these prioritized gene clusters. So in work package 4, we then look at the microbial compounds. So we identify the molecules. We very importantly also connect the genome to the metabolome so that we actually find the gene clusters that correspond to these molecules. So then we prioritize both the molecules and the gene clusters that specify their biosynthesis. And then finally, also within this work package, within particularly peptide antibiotics, we will analyze and we will produce analogs that will have better pharmacological properties and that will have hopefully better bioactivity. And finally, in terms of the experimental work packages, the screening and validation will be coordinated by Medina and also by Cons. So we have microbial and chemical libraries that are already produced or that are being produced within Marbles, creating compound libraries and strain libraries. We use high throughput screening to and connected to bioactivity assays to find novel molecules of bioactivity. And then we also optimize the bioprocess. So how can we optimally ferment these strains and use morphological engineering and strain engineering to make sure that we can optimize the process so that we can have a sustainable production process. And then in the end, obviously, validate the molecules and validate these for their application for clinical or agriculture or for agroculture. Work package 6 is on sustainability, society, and valorization. So we want to bridge the gap between the Marbles project, obviously, and the stakeholders, such as industry, policy, and society, in order to assess the environmental impact and sustainability of Marbles, to determine the societal value of the project, to assist with access and benefit sharing, ABS compliance, and biosafety regulations, link up with the policy scene, and provide science-based contribution. And in the end, we'd like to maximize the potential for societal and commercial valorization. That's work package 7 is on dissemination. Like I said, you already seen the beautiful YouTube movie for the public. Communication, IP protection, knowledge management and transfer, and stakeholder engagement. So look at effective dissemination, effective knowledge transfer, establish strength for relationship and connection with stakeholders and user groups, obviously the IP rights, and engage citizens in the concept of marine microbiome biodiversity to increase awareness and the benefits of protecting our marine precious resources. So expecting impacts are to support sustainable biodiscovery, develop novel exploitation methods, develop novel bioactive natural products, provide alternative to chemicals, which is incredibly important also because since 2018, 2019, chemicals in agriculture are very difficult to apply nowadays. increase public-private cooperation in biotech, increase the public knowledge, biodiscovery, reduce the pressure on wild populations, and promote environmental and economic sustainability. And then Marble's impacts towards the United Nations Sustainable Development Goals. So it's very closely in line with Sustainable Development Goal 2 with 0 hunger, 3 on good health and well-being, 14 life in the water, but also addresses many other sustainable development goals. So on behalf of the consortium, I'd like to thank you very much for your attention. I'm looking forward to collaboration and connection to the other consortia. And I'd like to give the word back to Kostas, please.